Unlocking the power of social activities to preserve cognition in care facility residents. In the bustling hallways of care facilities, a silent yet impactful revolution is taking place. A study conducted by the Amsterdam University Medical Center has unearthed a groundbreaking revelation. Social activities have the power to halt cognitive decline in residents residing in long-term care facilities. Imagine the realm of a care home, where every interaction, every shared moment, holds the key to preserving cognitive faculties. Professor Hein van Hout, a luminary in the realm of care for older people, spearheaded this riveting research endeavor. Motivated by a personal anecdote about his grandmother's cognitive descent, this journey began with a single question: could social engagement be the antidote to cognitive decline? The study, a meticulous analysis encompassing 3,600 patients across 42 Dutch and Belgian care homes, unraveled a narrative that challenges the status quo. Over 18,000 cognitive assessments were conducted, revealing a compelling correlation. Social activities act as a shield against cognitive decline, particularly in individuals with minimal impairment or those at the cusp of decline. Social activity, Van Hout elucidates, is the elixir that we have been seeking. From heartfelt conversations to communal outings, every interaction, no matter how seemingly trivial, emerged as a catalyst in preserving cognitive acuity. The study's revelations transcend mere scientific inquiry. They hold the potential to reshape care guidelines, redefining the very fabric of social engagement in care facilities. The implications are profound, echoing beyond the confines of care homes. A tapestry of possibilities unfolds: a reimagined approach to staffing, a surge in volunteer engagement, and a paradigm shift in societal costs associated with long-term care. Van Hout's vision stretches far and wide, envisioning a future where the essence of humanity, woven through social connections, becomes the cornerstone of cognitive well-being. As the sun sets on traditional notions of care, a new dawn emerges, illuminated by the promise of social interactions as the antidote to cognitive decline. The residents of care facilities are no longer mere recipients of assistance. They are active participants in a symphony of shared experiences, each note resonating with the melody of cognitive preservation. In the quietude of care facilities, a vibrant transformation is underway. The seeds of social engagement have been sown, nurturing not just cognitive acuity but a sense of vitality and connectedness that transcends age-old barriers. In this new narrative, the power of social activities shines brightly, a beacon of hope for a future where cognitive decline is not inevitable but an obstacle waiting to be overcome. Source: Uric Alert. Next story. Origins of life. Did soda lakes spark the first cells? In a groundbreaking study published in PNAS Nexus, scientists have put forth a fascinating proposition: did the first cells evolve in soda lakes? Imagine the serene and picturesque Last Chance Lake in British Columbia, Canada, being a modern-day representation of the ancient soda lakes that might have kick-started the emergence of life on our planet. The key lies in the unique chemical composition of soda lakes, characterized by high levels of dissolved sodium and carbonate species. These conditions could have been just right for the genesis of the first cells. Picture this: early cells potentially consisting of RNA enclosed within lipid membranes. However, a hurdle arises when we consider that RNA function necessitates divalent cations like Mg2+, which unfortunately disrupt primitive membranes made of fatty acids. Here's where the magic happens: the relatively low concentrations of Mg2+ in soda lakes might have provided the perfect balance, allowing both RNA and membranes to function harmoniously. In a quest to explore this tantalizing possibility, Zachary Cohen and his team ventured to Last Chance Lake and Goodenough Lake to collect water samples post-seasonal evaporation. What they discovered was nothing short of extraordinary. These soda lakes boasted approximately 1 mNA+ and 1 mmMg2+ at a pH of 10. In a series of experiments, the researchers observed spontaneous extension of short RNA primers in the lake water at rates comparable to standard laboratory conditions. Further adding to the intrigue, when fatty acids, believed to have been abundant on the early Earth, were introduced into the lake water, they assembled into membranes. The membranes formed even in dilute water, mimicking rainfall events, and remarkably persisted when surrounded by concentrated lake water from the dry season. This suggests that soda lakes could have provided the ideal environment for essential processes in protocell development. Picture RNA copying and ribozyme activity thriving in the dry season, while vesicle formation took place during the wet season, painting a vivid picture of the dance of life's origins. This study opens up a realm of possibilities, hinting at the tantalizing idea that soda lakes could have been the cradle of life as we know it. The synergy between RNA and membrane function in these unique environments sparks new questions and avenues for research, inviting us to re-examine the origins of life on Earth. So next time you gaze upon a soda lake, envision not just a tranquil body of water but a potential hotspot for the birth of the very first cells. The mysteries of our origins continue to unravel, painting a mesmerizing tale of life's humble beginnings in the most unexpected of places. Source: Uric Alert. Next story. Sweat protein's potential role in fighting Lyme disease. In a stunning revelation that could potentially revolutionize the prevention and treatment of Lyme disease, researchers from the Massachusetts Institute of Technology, MIT, and the University of Helsinki have uncovered a fascinating link between human sweat and a protein that may offer protection against this debilitating bacterial infection. Lyme disease, transmitted by ticks, affects numerous individuals in the United States annually, with symptoms ranging from fever and fatigue to more severe, long-lasting effects that can persist even after antibiotic treatment. However, a recent study published in Nature Communications has shed light on a protein called SCGB1D2, found in human sweat, that exhibits the remarkable ability to inhibit the growth of the bacteria responsible for Lyme disease. While the exact mechanism by which SCGB1D2 combats the bacteria remains a mystery, researchers are enthusiastic about the potential implications of harnessing this protein's protective properties. From developing skin creams to prevent Lyme disease to exploring alternative treatments for infections unresponsive to antibiotics, the prospects are both promising and exciting. Surprisingly, genetic variants of this protein have also been identified, with one variant showing decreased protective effects against the bacteria, potentially increasing susceptibility to Lyme disease. This unexpected discovery underscores the complexity of human genetics and its role in influencing disease susceptibility. Through groundbreaking genome-wide association studies and laboratory experiments involving mice, the research team demonstrated the significant impact of SCGB1D2 in preventing Lyme disease. Mice injected with bacteria exposed to the mutated variant of the protein became infected, whereas those exposed to the normal version remained uninfected, highlighting the potent protective effect of SCGB1D2. Moreover, the researchers' collaborative efforts extended beyond MIT and the University of Helsinki, with researchers in Estonia replicating the study's results, further validating the significance of these findings. As scientists delve deeper into understanding the antimicrobial properties of SCGB1D2 and its potential applications in preventing and treating Lyme disease, the future holds immense promise for novel therapeutic interventions that could benefit individuals resistant to conventional antibiotic treatments. In a field dominated by immunoglobulins, the revelation of secretoglobins as potential antimicrobial agents opens up a new realm of possibilities for combating infectious diseases. With continued research and exploration into the diverse functions of human secretoglobins, the scientific community stands poised on the brink of transformative discoveries that could revolutionize the landscape of disease prevention and treatment. As we embark on this exciting journey of scientific exploration, the protective potential of SCGB1D2 offers a glimmer of hope in the battle against Lyme disease, paving the way for innovative strategies to safeguard human health in the face of infectious threats. Source: Uric Alert. Next story. Brown University pioneers brain-inspired wireless system for tiny sensor data collection. In a remarkable breakthrough, Brown University researchers have developed a cutting-edge wireless system inspired by the brain to gather data from minuscule sensors no larger than a grain of salt. Published in Nature Electronics, the innovative approach opens new possibilities for wireless sensor technology, envisioning a future where swarms of inconspicuous sensors could be used in implantable and wearable biomedical microdevices. The sensor network is ingeniously designed to allow these tiny silicon chips to be implanted into the body or integrated into wearable devices. Emulating the sparse communication style of neurons in the brain, the sensors detect specific events as electrical spikes and wirelessly transmit this data in real time using radio waves. This method not only conserves energy but also optimizes bandwidth usage by sending only relevant data in short bursts, mirroring the brain's efficient data compression mechanism. By adopting a radio frequency transmission scheme, the system becomes scalable, addressing a prevalent issue in current sensor networks where synchronization is crucial for proper functioning. This breakthrough not only propels wireless sensor technology forward but also sets the stage for revolutionizing how data is collected and interpreted from these small yet powerful silicon devices. In a world inundated with sensors, from automobiles to workplaces, and even homes, the potential application of this technology, especially within the human body, holds immense promise. The researchers believe that this brain-inspired system could lay the groundwork for the next generation of implantable and wearable biomedical sensors, addressing the increasing demand for unobtrusive yet efficient microdevices that can map physiological activities seamlessly. The sensors operate with extraordinary energy efficiency thanks to external transceivers that wirelessly power them during data transmission, eliminating the need for batteries or constant recharging. This feature enhances their versatility and convenience, making them adaptable to various scenarios and environments. The team's meticulous design and simulation efforts have paved the way for the creation of these sophisticated microelectronic sensors. Leveraging previous research on neural interface systems, the researchers have achieved a significant milestone by demonstrating the system's efficiency and scalability. Testing the system with 78 sensors in the lab showcased its ability to collect and transmit data accurately, even when the sensors operated independently. Looking ahead, the researchers aim to further enhance the system's power efficiency and explore broader applications beyond neurotechnology, highlighting the potential for this brain-inspired wireless system to redefine the landscape of sensor technology. This groundbreaking research not only marks a significant advancement in the field of wireless sensor technology but also sets a new standard for harnessing the power of brain-inspired designs to drive innovation in biomedical microdevices. Source: Uric Alert. Next story. Managing CAR T-cell therapy complications, redefining the role of MRI and lumbar puncture. In the world of cutting-edge cancer treatments, CAR T-cell therapy has been hailed as a game-changer. But with this revolutionary therapy comes the risk of complications, particularly neurotoxicity, which can lead to serious neurological symptoms. Traditionally, clinicians have relied on MRI and lumbar puncture to diagnose and manage these complications. However, a recent study published in Blood Advances has challenged the necessity of these invasive tests, opening up new possibilities for more patient-friendly approaches. Led by Dr. Guillaume Manson from the University Hospital of Rennes, the study investigated the use of electroencephalogram, EEG, as a potential alternative to MRI and lumbar puncture in managing CAR T-cell therapy complications. The findings revealed that EEG, a non-invasive test measuring brain activity, could be a valuable diagnostic tool in assessing neurotoxicity, potentially eliminating the need for more invasive procedures. The research, which involved 190 patients undergoing CAR T-cell therapy, showed that abnormal EEG results were more common in patients with severe neurotoxicity. While MRI scans often yielded normal results and had minimal impact on treatment plans, EEGs played a crucial role in detecting brain dysfunction and guiding adjustments in medication. Dr. Manson emphasized that these results do not discount the importance of MRI or lumbar puncture but rather suggest that a more tailored approach is needed when managing CAR T-cell therapy complications. By updating existing guidelines based on clinical evidence, physicians can make more informed decisions that prioritize patient well-being and avoid unnecessary procedures. The study's implications are far-reaching, offering a potential shift in the standard of care for CAR T-cell therapy recipients. As further research is needed to validate these findings, the future of neurotoxicity management appears to be moving towards a more personalized and patient-centered approach, ultimately enhancing the quality of care for individuals undergoing this groundbreaking treatment. In a field where innovation is key, this study serves as a beacon of progress, paving the way for a more efficient and effective way to handle the complexities of CAR T-cell therapy complications. As we continue to unlock the mysteries of cancer treatment, the integration of EEG into clinical practice could mark a significant milestone in improving patient outcomes and redefining the standards of care in the field of hematology and oncology. Source: Uric Alert. Uric Alert.